We are recording today's webinar, so I want to welcome everyone this morning to the Soil Health webinar series for 2025. Our first January webinar here is on the topic of zooming into soil health through a producer, an educator, and a researcher's perspective. Just a few housekeeping things before we get started. We will have one CEU, 4 CCAs in the soil and water management category at the end of today's webinar. So if you need those credits, make sure you stay tuned to the end to get those credits. The session is being recorded and will be available on the Ag Crops team website and the OSU Agronomy YouTube channel. And also, the CEUs will not be available for the recorded session. Feel free to submit any questions that you might have during today's webinar using the Q&A feature at the bottom of the screen. The chat box at the bottom of the screen is what we are going to use to get feedback from the audience today. So you can also email me questions at cochran.474@osu.edu if for some reason the Q&A function is not working. And with that, we will get to our first interactive point here. So feel free to go into the chat box and make sure you're messaging everyone, so all panelists and attendees, and share what city and state you're joining from this morning. So today's speakers have sort of a different format. We did a pre-recorded panel discussion, and then we have two of our speakers from today that are going to join us for a live Q&A session from the audience. So the first is Mr. Ron Snyder. He is a farmer from Wood County, Ohio. He is near Pemberville, and you'll learn a little bit more about his operation here in just a little bit. Speaker number 2 is Dr. Alan Franzluebbers. He is a professor with USDA. And then we also have Alan Sundermeier, who is the director of the Conservation Action Project here in Northwest Ohio and a retired extension educator. So I am your host this morning, Rachel Cochran. I'm a Water Quality Extension Associate. Joining me, we also have Gigi Neal and Kendra Stahl, A&R educators for OSU Extension, Dr. Stephanie Karhoff, a field specialist for agronomic systems, as well as Sarah Noggle and Manbir Ricard. So with that, I am going to start playing our recording. If someone can give me confirmation that the sound comes through, that would be great.
Alan, can you share a bit about your background and the work you do as a Conservation Action Program Coordinator? What excites you most about working in soil health?
Well, after 26 years as an extension educator, I retired, and then I was approached by the Conservation Action Project to be their coordinator. So they're a 7-county area here in the Maumee River Basin. It's a group of farmers, educators, and retailers that are looking at innovative programming. We're looking at that to try to improve conservation practices here. Also, Vinayak Shidikar and I established the Healthy Soil, Healthy Environment Signature Program for OSU. Now, Manbir, you're in charge, I guess, of the soilhealth.osu.edu website that got established. So, those kind of things I think are getting us on the screen as far as Ohio State participating in soil health. So, this is work that I'm feel privileged.
Sorry about that.
To continue. So what's exciting me about it is there's a lot of unknown biological functions, and we're just getting to the surface of testing this and learning about it. I learned a lot from my times helping the organic grain growers. They really had to manage their nutrients and understand soil health, and now I'm trying to help the conventional farmers do the same.
Thanks, Alan. Yeah, thanks for setting the soilhealth.osu.edu website. That's been very instrumental and a great resource for the people out in Ohio and beyond. So thanks for really doing that. Question for Ron. Tell us about your farming journey. How have soil health practices shaped the way you manage your land?
When I started out on this, it was probably 15 years ago. The first 3 years were pretty tough. And when I look back upon it, yes, they were tough. But here's the deal, and this is one thing where I think a lot of people fall short, is when you are going from basically a chemical operation of fertilizers, insecticides, pesticides, those type things, and you're not focused on the biology side of it at all. You have no idea of the biology side. So when you start out with the first 3 years, those first 3 years are very important because you are trying to change your soil over to a biological format, so to speak, to get things to work. And so you got time in there that's you're fighting to get the biological going. And when I started out 15 years ago, there's nobody else doing this. And it was a struggle. Now I think what I know, I think we could probably crank that back pretty easily to 3 years, but you're going to have to follow a pretty regimented path to get through that. Now, where I'm going now, well, we're definitely into biologicals. No two ways about that. I've worked with biologicals now for about 5 years. Rhizophagy is the key thing to moving nutrients. You have to understand that. If you don't understand it, there's not much reason to do any testing. The newer tests that I use would be Haney, and I use 2 different metagenomic tests and then SAP analysis. The current soil test does not work. You cannot get a recommendation for fertility off of it. I don't understand why it's still being used, but I don't make those decisions, and that's somebody else. But you cannot do that. And then I have gotten into nutrient efficiency is kind of where I'm in between switching to another part of this whole gamut. But nutrient efficiency is extremely important. When your fertilizers are only 10 to 30% efficient, I'm not going to pay $1 and get 30 cents. Doesn't make sense. So you got to change that somehow to make your operation a little more functional, let's say. And then where I'm heading now is this is really kind of funny how this one came about, but I'm looking at wheat and I'm correlating cover crops to the weed pressure that I see in the field. That is what is coming up this next year. And what I'm trying to do is try to figure out the biological indicators on the weeds to see if I can pick up something that would be the same as the cover crop that we would plant. Yeah, that's where we'll leave her for now. So thank you.
Thank you, Ron. And now we're going to switch over to our other Alan that's with us today. And question for you is that you've been involved in some fascinating soil health research. What recent studies or findings are you most passionate about and why?
Well, thanks, Gigi, for the question. I'm most passionate about the current study that I'm doing, and this is really undertaking an assessment of root zone enrichment of soil organic carbon and nitrogen. This is a concept that is developing. It's got some publication behind it, but it's not widely known yet. So this assessment requires us to sample a little bit deeper than typical. And the idea here is that carbon sequestration studies typically require a timeline from the starting point of, say, today, and then 10, 20, 30, 40, 50 years from now. And that all takes time. And you can only really do so much because each management system is actually very different. And we know that management changes soil carbon and nitrogen too. So I'm really interested about how we can do this from just single probing of the soil. And I believe that it can be done because the soil that is underneath the top foot of soil is really not under the control of the land manager. The land manager really has an influence on the top foot of soil. And that top foot of soil, it can be changed, and it is changed. But what happens below that 1 foot of soil is actually something that's pedogenically derived. It's through the soil formation factors. And those soil formation factors are what was given to every landowner. They were not necessarily anything that due to recent management. So that sets the baseline. And that baseline is very useful then to calculate, well, what is the land management effect over the history of a farm? And the history of the farm, we have to kind of define it as approximately a generation of a farmer, or even say 50 years. Because we know that there's a legacy effect of our management on the soil properties. And some soil properties are changed below 12 inches, but those are typically chemical properties. And then the resident physical properties, which won't necessarily change, but the chemical properties could change. But the organic matter properties really are only changed in the top foot of soil due to management. So that's what's exciting for me on this aspect. And certainly the management variations that occur on each farm are important. And so we can do this, get a lot of information from a number of farms just by sampling soil with this root zone enrichment calculation approach. So it requires, technically, it requires at least 3 depth increments. Typically, I'm sampling 0 to 4 inches, 4 to 12 inches, and 12 to 24 inches. And that's kind of a minimum. It'd be nice to have more depth increments, but we don't necessarily need to go so much deeper, and we don't have to necessarily have that many more depth increments, because naturally the soil organic matter properties are following this nonlinear equation. It's a logarithmic equation that typically is generalizable across many different soils. There may be some exceptions, but it's in general more than 90% of the fields will follow this example. So that's what's exciting me right now, and I look forward to the other questions.
Pretty sure that would generate really good information, and I bet that's a lot of work digging deeper into the soil, right? Question to you, Ron Snyder. On your farm, what specific strategies have you found effective for nutrient management while improving soil health? And if there are some lessons learned during this journey that you want to share with our audience, that would be great.
Well, I kind of alluded to a couple things previously. There's two drivers from what I am seeing from my vantage point, and that is a microbial diversity with the functionality of that microbial diversity. Those are the two key takeaways. It goes right back into rhizophagia again. Now, if you understand what rhizophagia is, it's relatively simple, but you need to understand the concept of it to make the following tests work, 2 metagenomics that I use. And when you actually get into the metagenomic testing, what you are seeing is a microbial community that is there, and what we're looking for is that microbial community functioning properly to bring the nutrient from the soil to the plant. Once I started to understand that and how that functioned and worked in tandem with one another, I haven't used phosphorus and potash in 10 years, and there's no reason to be using it, quite honest. And I turned around and I used a SAP analysis on a test that I did here last 2 years ago now, I guess. I had corn this year, it was wheat the year previously, 2 years now. And when I started out with that wheat field, of course I take Haney, and I found out that it was telling me I had 160-some pounds of available phosphorus available in the soil for the plant. Well, if there's that much soil that's there and that soil has that much ability to produce that nutrient, why would I put it on? Because when you put on a nutrient, example, let's put P on, you shut down the rhizophagy process. So now you're going from using a fertilizer-based nutrient, what word do I want? Condition to a biological condition. And when you put the fertilizer on and the plant gets the fertilizer, it's not going to exudate for phosphorus, for example. On copper, it's not going to exudate for copper. And that's how this system works. So the more fertilizers you put on, actually you're shutting down the natural system in the soil that can save you a bunch of money. And that's how I see it. I've used now, and then I used the second generation of metagenomic testing also, which that one I'm still working with. It's a great test. I really like it. And where I've really used that is on biologicals. I've tried biological for about the past 5 years. I can't get a yield increase out of them. They're just not there. So I started on this other metagenomic test, and as I come to find out, I got a call from one of the owners. He says to me, "What in the world are you doing up there?" He says, "You have some of the most diverse soils we have seen." And I think I was running like in the 70th percentile on his diversity. And so as you go through it, now we just got to get the diversity to function to the plant to bring this around to make the whole system work. There's your nutrient efficiency. Rhizophagy is roughly 100% efficient. It's a natural process. And we circumvent that by using pesticides, insecticides, fertilizers, all the synthetic inputs. So yeah, that's kind of where you can get some efficiencies out of it.
Thank you, Ron. Alan Franzluebbers, your paper on adjusting nitrogen fertilizer based on soil test biological activity presents an exciting approach. Can you elaborate on how soil indices might lead to better nitrogen management and what growers should know about implementing this?
Yeah, thanks for the question. I use a test called the soil test biological activity, and it's similar to the Haney test. Dr. Haney and I went to graduate school together, and although I use a different approach, a slightly different approach, it's based on the same concept. It's a simple, standardized approach to describing the ecological status of soil. It relies on the organic resources that are in soil. And typically, those organic resources are depleted under a frequent tillage regime. And that's just the nature of our agricultural system in the current paradigm. When soils are frequently tilled, they just are losing their organic resources. On the other hand, if you use a conservation approach, using various agroecological approaches, you can build up the organic resources in the surface soil. And it would be great if you can build up the organic resources deeper in the profile. But actually, that may not be possible, and it may not be desirable because that could lead to environmental degradation as well. So the organic resources that are accumulating near the surface are really important. And we should learn and that would be nature's way of doing things. Nature, all natural systems also have this organic accumulation at the surface. So these organic resources are building up at the surface. So that's an important first step. So the soil test biological activity, the way I do it is determined from the flush of carbon dioxide released during the first 3 days of incubation following re-wetting of dried soil. So we dry the soil so that we have a soil condition similar to any soil sample sent to a soil testing lab. And that's a requirement that actually is a good thing, because that preserves the condition of the soil. It doesn't allow microbes to interact with the conditions in the soil during transportation or however it is handled. So that's the first step. So we use a sieve sample. We sieve rather coarsely also. This is an consideration because a lot of times soil testing labs like to have very finely ground pulverized soil, which is useful for homogenizing, getting a good sample, representative sample, but actually it leads to some problems in the laboratory when we try to estimate biological activity, because essentially water is not really infiltrating properly. We get ponding of the water even in high organic matter soils, and then we have some discrepancies that we look for. So we do coarsely sieve. Okay, there are other ways of estimating biological activity as well, and all of those approaches can work, but they need calibration. So I've conducted field trials, approximately 150 in corn trials in our southeastern region, and approximately 90 in fall stockpiled tall fescue. These trials led me to the conclusion that really we can use this biological activity test to change the nitrogen fertilizer recommendation. The current paradigm assumes that all of the nitrogen that is required by a crop needs to be applied in a fertilizer form, whether that's inorganic or organic, that we need to supply this nitrogen so that the plant can take it up. And that actually is the result that I get when soils are really low in biological activity. That's the recommendation that I would make also. Low biological activity, use the traditional paradigm. But if you're starting to increase the biological activity with agroecological approaches that preserve organic matter near the surface, then the nitrogen requirement is actually reduced because there's nitrogen supply by the soil itself. The organic resources that are there are relatively available, and they can lead to less need for nitrogen and fertilizer. Now, the system isn't perfect. If it were perfect, we would have probably discovered this 100 years ago when we first started the research. But thousands of researchers and at least a century of research has really not led us to the same conclusion that we still have, is that we have a crop with a nitrogen need and we apply that. And that's led to over-application in many cases, maybe not in all cases, but certainly in some cases it's led to over-application. And then, so we have a better way. We know that the soil can be changed and the organic resources are important. So this biological activity test is one way to estimate that and can lead to less need for nitrogen fertilizer. That means that farmers can save money, they can increase their profitability, and the environment isn't exposed to the high levels of nitrogen that we've been accustomed to. So it's an active area of research. There's certainly some resistance in the scientific community for the idea, but actually the concept is sound. It's logical. It makes sense. If we can increase biological activity, then and the reason that I can say this is that nitrogen mineralization, the formation of inorganic nitrogen by soil microorganisms, it's a biological process. So as long as we estimate that relatively well, then we can do that. Now, the complication comes in that the calibration studies are necessary because the environmental conditions are different every year. For every field, the temperature fluctuates, whether it's cool in spring or warm in the spring, and the rainfall is different. So those factors control biological activity. So the biological activity test is really only a potential, and so we are not perfect in our estimation. But it's an exciting area of research where we can actually improve nitrogen efficiency just by understanding the biological activity. Thank you.
Those are great points, Ron Snyder and Alan Franzluebbers. I think the conversation is steering towards that. We really need to be paying attention to how our soil biology is working, so we can really manage our nutrients better. It looks pretty good when we are talking about that we're going to increase biology so we can decrease nutrients, but when it comes to like whether on-farm practicalities or in research, we try to do calibrations and trying to provide some recommendations, then it all gets complicated. And on top of that, of course, as Alan mentioned, weather is a big deal when we talk about biology and how that works. Related question to you, Alan Sundermeier. We have heard from Ron and Alan that these practices can work. We should be really paying attention to soil biology and soil health indices. But as someone who connects researchers and growers, what are the biggest hurdles you see in adopting these soil health practices, or how can these challenges be addressed to foster further collaboration so we can come up with better ideas and better research and better practical things that can be adopted at the farm?
Well, farmers don't like change. You know, they're used to what they've done for years and what their dad and grandfather did. So when you say, you know, to take on these new soil health practices, there's some risk. You know, there's some learning involved, and you don't want to go too fast with some people. They'll make mistakes if they don't understand what they're doing. But I think, you know, we have to look at the economics. You know, we're going into a downside now with farm economics. So when you talk about, well, you have to spend money on cover crops, you have to do this or that, there might be, you know, a loss of yield the first few years. Farmers are going to be questioning, why should I do that? And, you know, we need to connect the dots better to show them that this is possible. You know, when you're talking about risk, there's ways you can manage that risk, and you have to get the information to the farmers to make them understand that it still is worthwhile to take this risk on. You know, when you're trying to connect researchers, they have, you know, a lot of big ideas. The grower is concerned about his profitability on the farm. So you have to keep things practical, and you have to make sure maybe he doesn't do the whole farm all at once. You know, take a section of land, maybe not up to the road where all the neighbors going to see it, and, you know, do some experimenting. If you go a whole hog and you make a mistake, you are setting yourself back, and there's some problems. We need to find, you know, good peers. And you look in the communities, you know, around Ohio and elsewhere, you may have some good, outstanding farmers like Ron to follow. You may not. If you can find a peer that you can follow and talk to, you know, that can help you overcome some of these challenges. So you need to look at maybe establishing some long-term data. You know, as we look at the benefits, they may not be there the first year or two. So we look at places like Ron's farm that has long-term good soil health practices and try to get the farmers getting established new in this to understand that this is possible, and this is the result of what you can achieve. So by having some mature soil health farms to look at, and maybe field days, you know, get farmers to see that it is possible in their local environment, and not looking at, you know, the ultimate research that may not be even close to where they're at, to the soil type and weather changes. You've got to localize these things to get those local farmers to understand it, I think. And how do we do that? Well, now we have a lot of farms, the capability with technology of using yield monitors and getting more data intense. You know, data-driven farming, I think, is where we're going towards more and more to understand our soils. So why aren't we doing more research on our own farm, looking at our own equipment, our own situation, and starting to look at some comparisons? It can be very easily done. You know, that's a lot of what our Conservation Action Project is doing, simply taking maybe one or two things and comparing them and getting some good results using good statistics, getting replication, randomization. We can teach farmers that are new in this to set up their own farm trials and get some data that means something to them on their own conditions. And that's where the researchers need to get out and be on those farms and help the farmers with the first step. Once they understand that and can properly set up that on-farm research, then they're off and running. They'll look at all different kind of comparisons and they'll understand it and they'll believe it. If they do it themselves, the believability and acceptability of those practices will be much easier than maybe listening to someone that they don't even know. So I think, getting to the practical side of things, maybe overcoming risk with some of our programs like H2 Ohio, farmers that collaborate with, cost sharing on some of these risk averse type things that can help. It may not be, a total answer to avoiding risk, but if you get some income to offset that risk, I think that helps too. So we have a lot of farmers, paying attention to these programs, but they're not maybe getting the education to go along with that. They'll take the money that helps avoid some of the risk, but what are they actually doing to follow those practices long-term on their farms? That's something that I think we're failing with, and the university and the government needs to do a better job of educating and maybe, withholding funds until they know that those farmers are able to understand what there's capable there and not give all the funds out without any responsibility back from it. So, a lot of money's been spent. What has been the reward from that money spent by the government? Sometimes it's questionable. So we need to have some accountability there. And the researchers can do that by getting the farmers to do those proper practices for soil health that will, accomplish our goals up here, which, is to reduce nutrient load into our rivers and lakes. So, it can be done. It's not rocket science, I think. We just got to go about it and take the time to do it. And, establishing those relationships is a lot of it, I think. So thank you.
Thank you, everyone. It's all great information and resources that our clients can take and hopefully put into practice. Now to all three of you, and we will start with Alan Franzluebbers, but the question goes to all three of you. For someone new to soil health, where should they start? What's an easy, impactful first step that can show meaningful results?
Okay. The question on soil health, where to start? Well, I think the idea is that you have to be convinced that change is necessary. If you want to go from a conventional system where you have commodity driven and the low prices and the high input costs that are there, then you would have to just get bigger. And that's the model that has been proposed and undertaken in many cases. Soil health is something about understanding the environment better, understanding the community of people better, and understanding how we can interact effectively. So soil health is the first step is to change the mind. That's why from a physically based scientist, that's maybe not what you expected, but that's I think what is really necessary is that understanding how plants grow, understanding what livestock do on the landscape. All of these things together are really important. So the first steps, though, once you understand that you want to have change, then you're going to then try to mimic nature. The mimicking of nature it means that you have to capture as much sunlight as possible. You have to use water effectively. You don't want to see your streams filled with sediment and nutrients that lead to degradation. So that means that you have to have plants that are robust, that you can harvest them appropriately. So obviously, I think in my view that the first steps are really consider perennials. Perennials mean that you don't have to disturb the soil as much. And that would be a good first step. That means that also the organic resources are going to accumulate because perennials leave behind a lot of residues, a lot of leaky roots that are depositing carbon and nitrogen behind. So I think getting perennials into the system would be great. I think that I would just say that perennial based cash cropping is something that we need to consider in the future. Because that's the way we're going to really jumpstart soil health and accumulate carbon. Soil health is based on the organic resources in the soil. If we don't have carbon resources, then we won't have good soil health. It's just as simple as that. I can let others comment further.
Alan Sundermeier, your
Well, of
thoughts?
course, my background in education, you never get enough knowledge. And if you don't understand what you're going to be getting into, you can make a lot of mistakes. So how do we get this knowledge? Well, you get it from fellow farmers that are successful. The Nature Conservancy has a farmer advocate program. And that's, I think, a good thing, where these farmers are willing to share what they're doing. They're doing soil health practices, and that's always good. We have the Conservation Tillage Conference, March 11th and 12th at Ada, Ohio. There's a lot of good sessions there. You can look at ctc.osu.edu. So that, finding mentors at these conferences, it's amazing, the hallway talk between speakers may be more important than the actual speakers sometime, but whatever you can do, to try to gain that knowledge, I think that's the first step that we can do. So, we've got to look at systems that people can understand. NRCS has 4 basic principles of soil health. You've got to reduce the tillage. Now, maybe that means instead of 4 or 5 trips of tillage, you reduce it down to 1 or 2 the first time. Then maybe the next time you go no-till. You might not be able to do it all at once, but you got to somehow reduce that soil disturbance. You've got to keep the soil covered. So by reducing your tillage, you're going to have more residue, and that's going to help protect the soil and build your soil health. So we need to have, as much as possible, keep some living roots in that soil as long as possible. And that's where cover crops coming in after your grain crop is harvested. In some of these mild winters, maybe not this winter, but some of our mild winters, we've had living roots almost the entire winter time. And that really helps the soil health keep established. And we got to look at, I think, more wheat and small grains in our rotation. We're doing a lot of soybean back to soybean, maybe putting corn in there. But there's so much more benefit you can get when you have wheat or small grain, getting a cover crop established early. It just helps with that rotation. Again, those principles of not abusing the soil, and that wheat, when you don't do tillage, so many guys, they'll harvest the straw, which again, you're losing some nutrients, but if you can no-till some covers right into that stubble and let that grow and get some really good legumes and other things established, then go back to corn, and you'd be surprised at how much that can really add to the soil tilth and the nutrients you can gain from those kind of systems. Why aren't we doing it? Well, economics. They think, wheat's not making enough money. Our ag economists to show long-term rotation effects by what you gain from the following crop can maybe offset what you're not maybe getting on a small grain income. So there's some things there that I think, as we're starting out, we can look at our systems and improve some of these things, I think, as we go along. Thank you.
And now to Ron, what are your thoughts?
What are my thoughts? I guess I'm going to look at it straight from the farmer's aspect. Number one thing, if I was starting over again, and I know what I know now, the first thing that, and I've told this to guys, go out and get yourself a Haney test. Number one, you have to have a basis of where you are starting. The second thing you should be doing is basing your seeding rates off of that test, because what you are trying to do is to increase the microbial community. Let's just stick with that one right now, not AMF. And if you're going to go out here and you have a field, and I have seen Haney tests come back 28, 32, 36 on CO2 burst, and you run from a range from 0 to 200, your ability of that soil to take on nutrients, you can do it. You can go out there with a full 60-pound rate, be prepared to go backwards, because you're not, this microbiome, the carbons, and everything needs to be built slowly. And as it keeps coming up, then you can up your rate and start playing around with what you want to do. You have to have that microbiome working first. And when you get that working, that's in direct correlation with the cover crop that you're planting and the amount and the rate. There's where the thing falls. I've tried when I first started out, oh, well, heck, you know, bushel of rye to the acre, 56 pounds, that's nothing. I had my own rye, we'll put on 2. Well, okay, that didn't work so good. Why didn't it work so good? And you got to go back and start asking yourself questions. And you're getting into something else too when you start in its microbiome world. You have to understand if you're going to go out there and you're going to place a diverse cover crop, a high seeding rate, and then you come back and you plant corn, be prepared to take a 10 bushel the acre yield loss. Because what is happening there is the corn is growing, you have this high microbial community. The microbes eat first. The corn will give up its energy and its sugars to feed that microbial community. And if you're starting to do that, you're going to take a 10 bushel yield loss. Real simple. I've done that. I've had that situation with biologicals I play around. You know, you put the biological out there and you think, okay, you're really going to get this yield bump. Not so much. You could go backwards real easy by not understanding the functionability of the soil, where you're at and what you're doing. And that's why I come back and say, you need to test first, find out where you're at, consult with someone, and then move forward slowly. Thank you.
Thank you so much. Those are great thoughts, and I think a great direction for our audience to get started with soil health, mainly understanding their system. So we're gonna open up the floor for the audience questions now. Audience can jump in, put their questions in the Q&A section, and we will start taking those now. Thank you.
Okay, so with that, we will pivot to the Q&A session. I'm going to stop sharing my screen here so we can have more of a gallery view.
Well, good morning, everyone, and I'm glad we could have everyone here today. We do have a question to start us off, and it goes to Ron Snyder. The question is, how is Ron estimating a return on investment for the test and products being used on his farm? You're muted, Ron.
Now, can you hear me?
Yep, you're good now.
Okay. First part of that question, how do I pay for the testing? That one's a real simple one. Number one, in the savings of the fertilizer that I use. That really, if you're going to use a Haney test, you're looking roughly $60. How much fertilizer do you need to not put on to make $60? It's a no-brainer. That one works real easy. The products I'm using, as far as the biologicals go, and that's where I'm going to go with the question, because I'm not quite sure I understand the question. But the biologicals, that one's a hard one to make pay. I'll tell you that right up front. I've been playing with that for about 5 years now, 6 years. And again, it goes back to the Haney test with that CO2 burst number. And you can actually push that further if you want to know exactly what microbes are in the soil that's actually promoting your nutrient efficiency. That's where I think we need to go with everything, is nutrient efficiency, not putting it on all the time. And the paying of it, I can make it pay real easy, a lot of the stuff, because I'm just not using a lot of the synthetic fertilizers. I don't use insecticides, don't use fungicides, I don't use treated seed. I'm no-till, cover crops, try to cut everything the way I can. So yeah, that's my answer, I guess.
Okay, the next question, and it could go to both of you. What role does manure have in the systems you are talking about this morning?
I guess
Alan, you want to start?
I can start, I guess. Manures, there's lots of sources, of course. So if you're looking at liquids, you need to plug the tile to get the nutrients to stay in the soil and not wash on through. You're looking at, short-term active carbon build up immediately from nutrients in the manure. And also if you have like straw or other cellulose in your manures, you have long-term carbon buildup. So these sources of manure are both immediate and long-term, but the question usually is, well, how much can I compensate reducing nutrient through fertilizer purchase when I use manure? And that is the challenge. Just like anything with soil health, how do we estimate these things? Well, you have to look at doing some soil tests, like your PSNT, your pre-sidrous nitrate test shows you the amount of nutrients in your soil. So if you're using manure like in the late summer, early fall, the year before corn, you could lose a lot of that manure nutrient over the winter through rainfalls and the warm winters we're having. So you can't always assume all that nutrient's going to be there when you need it, but it does help improve short and long-term carbon buildup, which then cycles and releases. So manure can be tricky. You need to do the testing to know where you're at with it.
Ron, do you have any comments?
The only thing that I'm going to say about manures, I think, is I've pulled this out of my readings. All manures need to be composted. To get the breakdown of it and get it into a more of a carbon source to begin with, the liquids can move too easily. I got a few chickens I raise and things like that I put on the garden, and I compost everything. And when you compost it, it really changes how it affects things and works. I don't have livestock anymore. I used to have livestock with liquid and solid. I don't want to get into it because I got some very strong opinions about this. But yeah, it needs to be looked at, and I'm going to stick with all manures need to be composted. Thank you.
Do any of you, either of you, have experience using Johnson Sioux bioreactor compost extract?
Well, the Conservation Action Project worked with a farmer in Defiance County, and we had several plots out. And I think, that's a homegrown source of biological. You're putting your own ingredients into this extraction process. We're trying to increase the fungal content of our soils. So you're looking at your own fungal base from your own soils. We're just starting the research on it. Like all biologicals, there's a lot of variations of things. But if you're going to be purchasing biologicals, I think this is a natural thing to be looking at. It's your own source. It takes some management to get it accomplished, but I think there's something to it here.
Is anyone researching the difference in nutrient quality of crops harvested from soils that are more biologically active? I guess this could go to anyone on our panelists.
Yeah, Ron?
In all my readings, it comes out of the research at the Jena Farm out of England. And any cover crops that you put out there, they've known this since the beginning of right before World War I, that crops raised naturally do have more density to them as opposed to crops with fertilizers. And when you start to play with this, and I'll just throw in this anecdote, I do a lot of different crazy things in my garden, so to speak, and it's all natural. But if you've ever seen plants fight off Colorado potato beetle, it's quite amazing to what they can do if the soil is healthy and it is biologically active. That is where I have seen my most movement with using biologicals. It's not in yield. It's not in yield at all. It comes from disease resistance is where I'm starting to really see it. And you're not going to get this with a common test, but the higher the CO2 burst number is, of course, higher the phosphorus number is going to be availability to the plant. But with the higher CO2 burst number, what you're starting to see, and I can speak to fusarium on this one, I had fusarium on one of my tests that was a high, very high, and where the biological was, that fusarium had totally changed and was not, it was very low. So what, and you got to go back to what it's fusarium after. Fusarium is after the microbiome or bacteria to eat. Once you start to feed the whole system in the soil, so you've got that complementary cycle going, then you don't have a lot of the diseases that bother a lot of people. Thank you.
Ron, this next question goes to you. With not using treated seed, what is your replant situation as well as when is your planting date? Why do you not see value in protecting the seed prior to emergence?
Okay, as far as the reseeding part, I've never reseeded with not untreated seed. Corn, soybeans, wheat. Where I am looking at this from, and it's probably a little different, you have to understand that each seed comes with a microbiome of its own. It is anywhere from roughly 2 billion on the outside of the seed and anywhere from 4 to 6 billion on the inside of the seed. Those are living organisms that can be triggered to start from dormancy in the seed itself. And what I have noticed is when you put a treatment on, what you're starting to do is you have that insecticide, fungicide sitting right there on the seed. And when you put that into the ground, now you have a zone around that seed that you're not going to be getting your arbuscular mycorrhizal fungi to attack that root and colonize that root and give you nutrients. That's exactly why I went away from the treatments on the seed. The replant, and I don't plant late, I'll plant early. And the whole key to the thing is understanding the biology. The Haney test is the lowest test I use. I'm into metagenomic testing, and that one is wild. I'm currently working with a company, and it's kind of funny on this one, but I'm working with a company, and that one is now we're testing the biologicals in my soil. We're testing the seed that is going into the soil, and then we're going to start looking at what biologicals can I use to increase my yield, because I can get the disease resistance, but I want yield. So that's kind of where that whole thing is going. Thank you.
Okay, are there any gaps in our scientific understanding of soil health that need to be investigated in order to move practices that improve soil health on farms forward? Alan?
Well, there's always gaps. I mean, it takes money to do research, and we need more long-term research because the soils change so much from year to year. You know, the biggest gap is trying to get consistency maybe in our results. You know, we can do Haney, we can do bee crop, there's a lot of science in soil health testing. And I think the gap is interpreting this very specific information and data that these tests give us into a practical application of that information that farmers can then either cut back the nutrients applied, you know, cut back on tillage, but we can now have proof that these change of practices are doing some positive changes in our soil. Are those positive changes resulting economically in improvements to the profits of the farmer. So there's the economists, the soil scientists, and the biologicals all need to come together into one, I guess, like case studies more so to get all these connections made. You know, the gaps are we have too many departments. We have to streamline this to get a whole farm understanding of what's going on.
Ron, I have 2 questions for you. How is Ron taking his Haney test? Is he using grids and pulling a test in each grid, or does he sample by type, by soil type? And then the other portion of that is, what's the best time to do the Haney test?
Okay, how do I sample? It depends on the farm I'm on. If I'm on where I live, I have one soil type from one end of the farm to the other, and it doesn't change. So that one is one sample for about every 60 acres. As far as when it gets into another farm with varying soils, with sands and this type of thing, I do 2 on that when I hit the sands, and then I hit the heavier ground to kind of see where we're at. The other part of it is, what was the other part of the question on that, Gigi? Do
What's
I
the
grid
best time?
sample?
Yeah,
Oh,
do you grid sample?
no, I do not. I don't look at nutrition the same way everybody else does. Nutrition is a biological component that we raise in the soil to feed the plant. It is not a nutrient put on. You have to understand that when you put on a nutrient, the plant shuts down for exudating for that nutrient. So by putting it on, you're actually taking and making the microbiome in the soil weaker rather than stronger. And this is a proven fact. I'm a bee crop advisor, so I get into some meetings that are really kind of unique, and it's really interesting to listen to them. And yeah, that's kind of where I'm at with it. And the tests, yes, they're a lot more expensive, but do they pay? Yes, they do. Thank you.
And we are getting close to our time, and we are not going to be able to get to all of the questions that are within the Q&A in the chat today, to be respectful of everyone's time. We will take the questions, though, and send them to our panelists, and hopefully be able to get those back to you in a timely manner. Rachel?
Perfect. Thank you, Gigi. I did just see a message come through asking Ron where your farm is located, so I'll answer for you if that's fine.
That's fine.
Ron is in Wood County, near Pemberville. So the CCA credits are currently up on the screen. I will circle back to those at the very end of the webinar. But we do have 3 more webinars in our series. So our next one in February is going to be understanding the basics and practicalities of biologicals. In March, we'll be talking about what soil tests tell us. And in April, we'll be talking about soil health in the urban sphere. So if you haven't already registered for those other 3 webinars, the link is the same, go.osu.edu/soilhealthweb. As Alan mentioned in the recording, we do have the Conservation Tillage and Technology Conference coming up here on March 11th and 12th in Ada, Ohio. So all information about CTC and any other Ag Crops programs will be available on the Ag Crops website. We will also be sending out the recording to today's program through email. And also, our 2024 eFields report was just released. You can find that at go.osu.edu/efields. As with our previous years, this document summarizes research results from on-farm research trials all across Ohio. So you can also scan that QR code to pull up the digital report. So thank you for attending. If you have questions after today's webinar, feel free to contact myself or Dr. Manbir-Ricard, and we will get any unanswered questions to our panelists for today. We will send out their contact information and their answers to those questions in our follow-up email. So with that, I will go back to the CCA credits slide for anyone who might have missed it, but thank you all for joining us today.